Okay, so we're going to be hitting the chapter on photosynthesis in a week or two, but that's okay. This lab will help us understand it a lot better. Okay, and I'm kind of excited about this. I've heard about this experiment before, and I've wanted to do it, and here I am. I found it. So what does this entail? Well, what's going on is we're we are investigating what photosynthesis does. And this chemical equation right here is a synopsis of what photosynthesis does. Okay, what does it do? It takes carbon dioxide. Let me make this nice and big so you can see it. It takes carbon dioxide plus water plus energy from the sun, and the plant produces sugar, glucose, and oxygen. One of the most important things that we can do, because um, all photosynthetic organisms, plants, algae, even bacteria, what they're doing is they're harvesting the energy in the sun and they're turning it into potential energy, which is sugar, for you and me, essentially. Okay. So what is happening with this experiment? Well, what we're going to do is we're going to take some leaf that has chlorophyll. That chlorophyll runs what's called the Calvin cycle. Again, we're going to hit that in a couple weeks here. And what that Calvin cycle does is it produces ATP, which is energy, following this motif right here. Okay, so again, it takes carbon dioxide and it takes water, and then it takes energy from light, and it makes sugar, and it makes oxygen. Okay, so we're making a solution of bicarbonate. Okay, that bicarbonate, what it's doing is in the solution is making carbon dioxide. So the bicarbonate is the source of the carbon dioxide that your plant discs are going to use in order to do this. And you're going to put energy in the form of light. Okay, so you're going to put a low light lamp 10 centimeters from a light. So you're going to put light, and of course the discs are in water, so it's getting water, and it's going to produce sugar. But this is not what we measure in the experiment. What we measure in the experiment is the fact that it produces oxygen. This is why it's important that when we put the discs in the syringes, that we degas them. In other words, we're taking all the gas from the atmosphere that's inside the discs, and we know that there's no more gas in the discs because they stop floating. Does that make sense? They come down to the bottom of the syringe. And then when we put light on there, the gas discs are going, forgive me, when we put light there, the discs are going to harvest that energy. It's going to take the carbon dioxide from the bicarbonate that we put in there, and it's going to produce sugar, and it's going to produce a gas. And as it produces that gas, that oxygen, is going to make the gas disc go to the top. So how many discs float should be proportional to how much energy we're making during photosynthesis, because oxygen is being made. Okay, so we're doing 4 different experiments. We're taking chlorophyll that has been exposed to the light and chlorophyll that has not been exposed to the light. So you have to think to yourself, which one of those mechanisms would be functioning? The one that has been exposed to light and is carrying out a function, or the one that has not been exposed to the light and is not carrying out the function? And it's not a difficult answer, but um, you have to have the answer because you're going to have to draw a hypothesis. Okay. So which one's going to function better? The spinach leaves that have been exposed to light or those that haven't been exposed to light? So that's one question. The second question is that we're using different concentrations of bicarbonate. So in one condition, this is low, the carbon dioxide is low, and in the other condition, the carbon dioxide is high. In which condition do you think more oxygen is going to be produced? Where the carbon dioxide is low or when the carbon dioxide is high? So essentially, this equation is an equation of equilibrium. And what this is telling us is that if we put something over here, it's going to make this equation move to the right. So the higher the concentration of carbon dioxide that you have, the higher the concentration of oxygen that you can end up with. Okay. So again, that's supposed to be part of your hypothesis. So that's the main part of this. Now, what we need to do is we need to make our solutions. Okay. So you're gonna need both beakers. Okay. And one beaker, you're gonna put 250 milliliters of water. You're gonna dump all of your baking soda that comes with your kit into it. Okay. And you're gonna stir it. You're gonna stir it as much as you can. And you're gonna see residual baking soda at the bottom. And that's okay. You want to see that because what that means is that you have a saturated solution of baking soda. And that's what we're going for. So once you have that saturated solution of baking soda, and again, how do we know? Because we have a little bit of baking soda on the bottom, okay? Now you're gonna take 5 milliliters of this solution and you're gonna put it into a second beaker. To this beaker, you're then going to add 250 milliliters of water to it, and that's going to be your 0.24% sodium bicarbonate solution, okay? Now what? Now you take your first beaker, you rinse it with distilled water, okay? And you're gonna put half of this 0.24 over here, half of it. And then you're going to add enough water to raise a total volume of 250 milliliters, okay? So to recap, we have 0.24% in here. We have 250 milliliters. We're gonna put half of that in here, 125. And then we're going to add water to a total volume of 250. So now this beaker has 125 milliliters of 0.24% sodium bicarbonate, and this beaker has 250 milliliters of of 0.12% sodium bicarbonate. Okay, so those are your two conditions, 0.24 and 0.12. Okay. Now you're going to need to make a detergent solution. So you're going to add 1 drop of detergent to 20 milliliters of water. And what the detergent does is it breaks the hydrogen bonds so that discs don't have to fight breaking hydrogen bonds in order to rise through the water. Okay. What else? Okay, so that's the so-called hard part. No, actually, I'm lying to you. You have to take your syringes, right? And you're going to put 10 discs of spinach leaves in there because that's what's going to be carrying out the photosynthesis. You're going to make the discs with the straws that are provided. Okay. So I gave you straws. So do the math. You have 4 syringes. Each one takes 10 discs. So you're going to need 40. Make extra. Make 50 discs. Okay. But you have to be careful. Okay. Half the discs have to be spinach that's been in light, and the other half of the disc has to be finished, that's been in darkness, because that's the other experiment that we're doing. Okay, so you're gonna put your 10 discs of leaves in there. You're gonna take your 0.24% solution, and then you're gonna go through the degassing. Okay, so what does that mean? Remember, you're gonna pull this in a vacuum. You're gonna pull in a vacuum, and then that's gonna take the gas out of the leaves, and they're gonna push it up. You're gonna fill it up with a solution. You're gonna pull in a vacuum, and essentially you're gonna keep on doing that. Your author tells you to do it about 2 or 3 times, and you know that you're successful when your leaves fall to the bottom. That means that all the gas has been extracted out of the leaves, and there's no reason for them to float anymore, and they're going to fall to the bottom of your syringe. Okay, so you're going to do one at 0.24. You're going to do another one at 0.12%, and then you're going to do the same thing again, but with the discs that have been in the dark. Okay. Let's see. Don't forget to add your detergent to the, let's see. We're going to add 6 drops of the detergent solution to the 0.24% sodium bicarbonate, and we're going to add 12 drops of the soap solution to the 0.12% sodium bicarbonate. Okay. Once your syringes are ready, okay, you're going to take your light source, and you're supposed to put it about 10 centimeters away. It depends what kind of a light source it is. Okay, be careful. If it's if your light source is LED and it doesn't produce a lot of heat, then 10 centimeters is going to be perfect. But if you bring in an incandescent lamp, it's going to create a lot of heat. And at 10 centimeters, you're going to kill your chlorophyll. Okay. So ideally, you want to find a light source that's LED. That way it produces light, but it doesn't produce heat. Okay. And as you do this, if photosynthesis is taking place, the discs are going to begin to float. And your job is to count how many discs are floating per given time. Okay. So that's the big picture. It's not very complicated. I'm looking forward to, I'm going to do this because I've got a couple students coming in tomorrow to do this experiment with me because they need help with it. And, you know, I'm excited to do it because I would love to see the discs float because they're producing oxygen due to photosynthesis. I mean, that is very, very cool. So please look at the video on degassing. The degassing is the most complicated part of this whole thing. And I hope that the overview helps you, okay? This is not a very difficult lab, but the author makes it sound very complicated. I mean, I know what I'm doing, and sometimes I get lost reading the instructions. So hopefully my overview helps a little bit, okay? So that's it for now. I hope that helps you a little bit. Thank you.